Ah, the coveted post-lunch spot. It's a beautiful place to be. So if you get bored, just go out and get another cookie. If you get really bored, get one for me too, and we'll be fine. Anyway, oh yeah, thank you. If I pass out, it's from low blood sugar. Anyway, welcome everybody. This is something that I hope makes a lot of sense. I'm compressing about, 4 hours of material into 20 minutes, so let's go. First, let's talk about the MolDX program. The MolDX program was started in 2011. One of the Medicare administrative contractors over California, Dr. Elaine Jeter, who's a pathologist by trade, saw that molecular testing was on the rise. And there was just no good way of identifying what was being paid for, how it was being paid for. And so she proposed to Medicare a statement of work. We'll call it a pilot, but it's really not a CMS pilot, it's a CMS statement of work, which basically said, CMS said that would be a great idea, but they said the MolDX program will use a methodology to determine if molecular testing is ready to be applied in the clinic using something called the ACE criteria, ACCE, that the CDC put together. Anyway, this is a graphic that I think a lot of you have seen, which is basically the CDC said when you're looking at any molecular test, you've got to look at 4 essential areas, and we've been talking a lot about that today, so I won't spend a great deal of time on this. Other than I think it was important to recognize that one of the things that even the Food and Drug Administration, and Dave said this in his presentation, clinical utility is not even looked at by the Food and Drug Administration. They look at analytical and clinical validity, but not clinical utility. And so basically, we were asked as the MolDX program to look at clinical utility of testing. Someone asked, well, what's the impact of the program? We're now in our third year. And what we have tried to do is try to say, let's make sure that everything that's taking place by the patients is reasonably safe and effective. And truly, it was a gap that was not currently being undertaken by any of the major payers. There was really no way of looking at molecular tests, and so it was kind of first in category. There has been a great deal of reduction of what I'm going to say less than appropriate testing or less than useful testing. And none of it was ordered by any physician or run by any lab in this room, by the way. You just need to know that. Also, but I think this is really important, I think people have not recognized that the MolDX program also has a mandate to help nurture molecular diagnostics. CMS, being reasonably forward-thinking, realized that there was going to be a need to try to facilitate this fledgling area if it was going to continue, and the MolDX program has that mandate. And what has the impact been? Well, after 3 years of this, Congress in the summer of this year said basically that HHS now has the authority, congressional authority, to choose between 1 and 4 MACs to determine coverage and or payment decisions for molecular testing for the nation. Now, CMS has not made that decision as of yet, and so truly we don't know how that's going to go forward, but basically Congress said we recognize that there's importance of using Medicare contractors with areas of expertise to help determine where things are going with the Medicare program. So let's talk about determining reasonable and necessary, or what we call clinical validity and clinical utility. You've got to remember that Medicare is not an insurance program; it's a law from the 1960s. And when they were thinking about medicine in the 1960s, think how different it is now, 40 years later. And this is something that when Medicare put together, they said, what are the things that Medicare will cover? These are the Medicare benefits. And one of the things that I think that a lot of people don't remember, that by law, Medicare can only cover certain things. They cannot cover a screening test unless it's been specified by law. That's in the Medicare statutes. It cannot do something that is purely confirmatory. If you already know something and repeating a test, and so they don't allow duplicate testing if you already know the information. Third, if it determines prognostic only, and if it's a purely prognostic test, then it's not covered by Medicare because it's not something you act upon. Third, if it measures the quality of a process, basically a quality control or a QC measure, that's not something that's covered by Medicare. It's considered part of the testing itself. If it's non-specified, basically, a screening for whatever. And if it's investigational, experimental, by available literature, that is where it becomes problematic, because who defines what is investigational or what is experimental? And you'll hear later on in one of my slides, we'll talk about how that is changing. Now, So if it fits into the category, is it a Medicare benefit, then the next thing we look at, is it reasonable and necessary? And we define that into two categories. Basically, is it clinically valid? Is it reproducible? Can you identify the effect you think it can identify? We've already talked about that. And then lastly, is it something that's useful? Can we act upon the information? Will we act on the information, and will it provide a benefit? And this is what is defined by clinical utility. And this is adapted from questions by Bruce Quinn and Felix Frew, and just some really good questions that I think are important. Now, I think this is something that we're all familiar with, and it's basically the hierarchy of levels of evidence. We have randomized clinical trials, which have a low bias. We have prospective interventional trials, which we'd call a traditional phase 2 trial. Basically, I'm going to take a patient that has a specific diagnosis. I'm going to treat them according to a certain methodology with some intervention, and then I'm going to have some type of outcome. Because there's no control, there is a medium amount of bias. And then we get retrospective observational trials. We basically take individuals and say, now we have seen what has happened with these individuals. Let's try to drill back down to determine why these individuals were different. There's a fair amount of bias in those individuals. And then lastly, we get case reports. And case reports, most people would say, truly, we don't know what to do with case reports. I mean, was it miraculous faith healing? Was it an unusual genetic mutation? Was it whatever, misdiagnosis? I mean, case reports are difficult to know how they fit into the whole information paradigm. This is the way usually standard of care is migrated. We start out with standard of care, which is determined by clinical trials. And Dr. Solid over there, who made the comment of, we can't let, what is it, Dave? We can't let good information get in the way of good science. Clinical evidence was horrible, whatever it was. I can't remember what it is. Standard of care is determined by randomized controlled trials, and then we get some information where we get new interventions, and then the standard of care migrates, and we change this. And this is something this is the way care evolves. Now, I do agree and readily admit there's things we do just because we do them. And there are many things that we do. We don't ever have information like this. They become historical. I mean, like digitoxin use in cardiac patients. How many years did it take us to finally show that it made a difference? The problem is there's been a certain push that has taken place with molecular oncology and molecular medicine. And it's been considered the holy grail. And what we're seeing groups are saying, I've got this standard of care, and now I'm going to throw in a few case reports. And what we're going to do is we're going to take most of what was out there, and we're going to completely abandon it and treat the patients according to the case report. And truly, there are many people who say, no, Dickson, this doesn't happen. Let me tell you, this happens. I have reviewed, as my MolDX program, I've reviewed many, 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 many data sets where the data sets are basically saying, because I've got this test, it can take person away from an ASCO or an NCCN guideline, and you could put them on a different pathway. And when we look at the data, it's this type of data. We've got case reports that are determining the switch to the standard of care. Now, I'm not saying this shouldn't happen. What I'm saying, it's very difficult. And most people would say this shouldn't happen, and yet we do see that there's at least that push to see that start happening. For a long time, as the MolDX program was reviewing everything that we do, we developed a group of subject matter experts. These are individuals that are working in the field, like oncology test oncologists, gastroenterologists, things like that. And what they do is they review tests. And the way they would review tests is looking at the data and say, is it something that is useful in the clinic? And there was a great deal of disconnect when we'd see a laboratory test that would come to us for review, and we'd start looking at the clinical utility part of the data. There was a disconnect because when we gave it to our subject matter experts, they would look at this and saying, well, there's no utility here. And so what we did is we said, okay, we're going to make the evaluation process something we call the clinical test evaluation process, not the same as CTEP from NIH, or, so just know this is an internal MolDX thing, and if you're mad at us, we'll change our name. This is what we do. We basically said it's very similar to some of the slides we saw earlier, saying let's divide information into categories that just make good scientific sense. A category 3A is a randomized controlled trial. A 3B is a prospective retrospective trial. And this is a very specific type of trial that looks at archived data sets that were part of an original clinical trial, and you were able to drill down using molecular markers into the individual arms of that trial to determine if there was a benefit. That's what happened with the Oncotype DX. That's how they got their approval. And then we get into other areas of 2A, a prospective observational study. And I like the term prospective interventional trial, where you're basically intervening in a patient. And in molecular markers, it basically says, okay, I've got a marker, I'm going to use that marker, I'm going to treat a patient in a specific fashion, and I'm going to determine an outcome on that patient. And then we have what I'm going to call 2B, which is what we call retrospective data modeling, which is basically looking at large data sets, trying to identify control groups and groups that were intervened in a certain way and being able to determine who had a benefit. These are very complicated data sets to look at, and you really need to have good data sets in order to make this take place. And then lastly, we have retrospective observational studies and then preclinical studies. And you'll notice that our categories level of evidence basically follow what the FDA uses for phases of trial. We did that on purpose just to say, look, we can understand these types of trial types. And what we said is, look, if you're going to go and you're going to submit a test for coverage to the MolDX program, we need to be able to determine, is this test reasonable and necessary? Does it have clinical validity and does it have clinical utility? And what we did is, after reviewing hundreds of different data sets, we recognized that there was a trend. And what that was is, unless a trial could come in with some prospective information saying we have a marker, we've used it in a specific way, we've treated a patient to an expected outcome, we had never seen a test that would be approved unless it had that information. Because you couldn't answer the question is, who should be treated and how should they be treated and under what circumstances, what outcome. And so we said, okay, if you didn't have at least a level 2A, a one-arm prospective interventional trial, we didn't think it would meet the category of review and or coverage. Now, there's some exceptions to that. We recognize that this molecular area is looking at new paradigms. And one of the things that we have heard criticisms over and over again is you cannot take molecular tests and treat them like drugs. The problem we face is, yet when I get a molecular test and it's telling me to take a patient and treat them with a drug or take them away from a drug, how can I compare the data of a case report to multiple phase 3 randomized controlled trials? And that's a problem. And that's a problem because, once again, even though we agree that it's hard to get those levels of information in molecular tests, we also need to recognize that when we're taking people away from well-established interventions based on limited data set, we can potentially really harm a patient. But yet we recognize that there's new paradigms that are out there. I mean, we've been archiving tissue specimens for how many years, hoping that there would come a day when we could use those tissue specimens and really drill down to what was causing disease. How do you deal with those archived tissue specimens? It would be an absolute travesty if we didn't use those archived tissue specimens to try to determine effects and try to determine what needs to take place. We also know that there are trials that will never be duplicated. It'll take too long, it's hard to find the patients. I mean, prostate cancer, early stage prostate cancer, trying to do a trial on early stage prostate cancer, do we have 20 years to wait to determine if a molecular marker makes a difference? Also, we've seen, as we get into smaller and smaller mutation types, as we get into more complicated mutation sequencing and other things, we are going to get into smaller and smaller data sets. So it's going to be unlikely that we're going to be able to really get some of these good data sets that we have traditionally used for determining if the test is appropriate. And then also, we also recognize that there's often a negative return on investment when it comes to laboratory testing and for many companies. And so we've got this idea where we've got these good scientifically proven standards of how data is collected and how data migrates, but yet we've got these new paradigms we've got to deal with. So one of the things that we have tried to push into all of CMS's lexicon is the old idea was that something was experimental and something was standard. And the experimental was determined by what was in the literature, and then all of a sudden something, you flip a switch and then it becomes standard. We all recognize this is the way it's going on. We have things that are experimental, we have things that are standard, and I'm using a term transitional, not translational, because translational has a experimental type, coming out of the lab, going into phase 2. And so I don't like the use of translational. I like the terms transitional. We've got markers that are translating out of the experimental reign into the standard treatment reign, and they're somewhere in between. And so what we are trying to do in the MolDX program is recognizing, even with our CTEP that we've been doing, is recognize that there are times when we are going to be in this transitional zone, and this transitional zone is where there should be some coverage that takes place. And determining that transitional zone requires a very high-level discussion with multiple stakeholders to determine what makes sense. This is a complicated graphic, and I'm not going to spend a lot of time. You can look at our website that'll go through all this information in detail. But what I wanted to highlight here is one of the things that is most powerful in our whole entire methodology of looking for coverage is we are establishing what I'm calling partner specialty societies. And what we have done is just recently, we're now in our second month, we've established a group of academic institutions, and they're all institutions that you would know. And we've got individuals that have agreed to sit as an advisory panel to the MolDX program. And this advisory panel will help us determine how do we bring new technology that is widely accepted into the marketplace. So we had a call with this academic group last Tuesday. What were we talking about? Introduction of next generation sequencing in a way that it would make sense to introduce to the Medicare population. Why? Because it's something that is important. Now, do we know how we're going to do it yet? No, but are we going to do it? Well, we're going to continue to evaluate it, and likely we're going to find a way of introducing it, because we've got this group of people where if a group comes to us of these independent advisors to say it's time to evaluate X, Y, or Z, maybe it's next generation sequencing this week, maybe in 3 years it's going to be proteomics, maybe it's going to be something else. But if they come to us and say, look, this is something that we think is valuable, then we believe that if we can go through and we can find a way that it either should be covered outright, or we can do something called coverage with data development or coverage with evidence development, where we can say it may not have and may not meet the evidentiary standards that we want, but at least if we can determine that it's important enough, and if we can standardize it well enough, then maybe we can start covering it and collect information along the way to get to the answer, is it something we should continue to cover or not? Now, I've kind of gone through this. Another thing that's important is that there are tests that we look at. Every test that comes into the door, we look at, and we have our executive committee will review it and ask the question, how great is the unmet need? How difficult would it be to get further data sets? Is this something that we can use archived specimens? And just recently, we've published 3 LCDs that deal with early stage prostate cancer, none of which met our traditional CTEP methodology. We recognized that this was something that we could never get the data to get to this area, and so it was something, after reviewing the data, we decided it could be covered, but it had to be covered in a very specific way in a very specific group of patients to make sure it was reasonably safe to do so. I talked about this. I'll only mention this. I talked about how the Medicare program absolutely believes, and when I say Medicare, I should say Palmetto, because I can't speak for Medicare, even though we're representing, but for Palmetto absolutely believes that we need to find a way of facilitating data access. We are proposing the formation of a group called the Molecular Evidence Development Consortium, and it's a group that we have involved many, many people. It sits along with Rich's and the ASCO's proposal is just a little different way of doing things. And this is not what I was supposed to talk about, so I won't, but bottom line is when we've presented this to our academic committee, there's incredible excitement of how this can bring forward to introduce new technologies into the marketplace, and that's all I'll say. Last thing, in the last minute and 20 seconds I have, is how do we deal with panels? That was a specific question. Here's the problem with panels, is that unless we can standardize information across a specific testing, we don't know what to do with things. We've heard from enough individuals that not 2 next-generation sequencing methodologies are the same way. And so how do we go through and say, if lab 1 is doing next-generation sequencing one way, and they've got a higher sensitivity and a lower specificity in lab 2, how can I aggregate that information to determine clinical utility? We also recognize that panels need to be disease-specific. I mean, for example, do we care if a colon cancer is ER positive? And the answer is no, because tamoxifen doesn't work in colon cancer. But we care dearly if it is in a breast cancer patient. And so the problem we're going to run into is we're going to find that certain mutations have great value in certain disease states, like BRAF mutations in individuals with melanoma, but in colon cancer, we know it's not a good target. It's a prognostic marker for colon cancer for the most part. We also recognize that there needs to be a hierarchical reporting, and that includes starting with standard, going to transitional, and then going to experimental. We believe that these panels will be something that will be continually adapting as we move forward. And we also need to recognize there's something that is, asymptotic cost. At some point, it's the same cost to do, 5 genes as it is to do 20 genes or to do 500 genes. But does that mean that we need to be looking at 500 genes and that should be paid more than for 5 genes or 3 genes? And so when we are starting to look at panels right now, we will look at them individually, but recognizing that this is a very difficult area, just like the FDA said. Thank you very much.
Thank you very much. Now we have Donna Messner, who's going to talk about CMTP's activity in molecular diagnostics and oncology.